In the last two lectures, we discussed about innate immunity and the barriers that are present in innate immunity. In this topic, I'll be discussing about the acquired immunity. Before starting this topic, I'll deal with some words for you here. The first one is immunophysiology and immunopathology. If I skip the word immuno, I'll discuss first physiology and pathology. What is physiology? Normal functioning of a body is called as physiology. And what is pathology? Pathology is something abnormal if happens, then the study of that is called as pathology. And basically pathology is a study of disease. So study of normal is physiology and study of abnormal, that is disease, is called as pathology. So what is immunophysiology? Study of immunophysiology is the study of normal structure and functioning of immune system. And on the contrary, immunopathology is the study of derangements in the immune system. See, if there is something that's gone wrong with the immune system, then that comes under the subject of immunopathology. Now the next two words are antigens and antibodies. See, what is an antigen? Antigen is mostly a protein. The first line is antigen is mostly a protein, and it is that substance which helps in the production of antibodies when it enters your tissue. Any substance which when enters your tissue, if that stimulates the formation of antibodies, then that is called as an antigen. Remember, most of the antigens are proteins. And now if I talk about antibodies, then all the antibodies are proteins. There is a difference. All the antibodies are proteins, but most of the antigens are proteins. And antibodies are the glycoproteins which are produced by the body as a result of antigenic stimulation. So whenever an antigen enters your tissue, your body responds by producing antibodies against those antigens. Like in this case, you can see a bacterial cell here. Now this bacterial cell has got multiple antigens over it with different shapes. See, a single bacterial cell can have different kind of antigens present on its surface. I have encircled 3 different kind of antigens present on a single bacterial cell, and against these antigens, specific antibodies are produced by the immune system. Now the next word is hapten. What is an hapten? Hapten is basically a non-protein substance which will never stimulate the production of antibody, but when this hapten, the non-proteinaceous hapten, gets attached to a proteinaceous carrier molecule, then that elicits the formation of antibody. Like in this diagram, you can see these are hapten molecules that are non-protein, and they will never induce the formation of antibodies, but when they get attached to a protein carrier molecule, then that forms a complete antigen. And now against this antigen, antibodies are produced. Antibodies are not produced against haptens, they are produced against antigens. So you can say that hapten is a non-proteinaceous component of an antigen. Now I'll start with the acquired immunity. As the word suggests, immunity which is acquired after birth. It is not present since birth. The immunity which is present since birth is called as innate or congenital immunity. Acquired immunity is not present since birth, and it is acquired after birth. The most important thing is that acquired immunity is present only and only in vertebrates. Non-invertebrates don't have acquired immunity. It is always generated whenever any antigen or pathogen enters your body and requires several days to get activated. See, innate immunity is always ready. Innate immunity is always ready, but acquired immunity takes time to get activated. Now I'll discuss about some unique features which are shown by acquired immunity. The first one is specificity. Specificity would mean that the products of acquired immunity are very specific. Like antibodies, if I talk about, antibodies are very specific to antigen. An antibody produced against the antigens of Mycobacterium tuberculosis, a bacteria that causes TB, will not act against the antigens of some other bacteria. So they are highly specific. The next is diversity. Diversity means there are vast number of antigens or pathogens against which your body can produce acquired immunity. That is, the acquired immunity recognizes vast variety of antigens. The third one is the discrimination. It is differentiation between self antigens and non-self antigens. Now listen to me carefully. Even our body has got some antigens which are self antigens. I mean, those antigens which belong to your body. I'll take some examples like the Rh antigen which are present on the membrane of RBCs, which are responsible for Rh positive and Rh negative blood groups. That is an antigen which is a self antigen. Next is blood group antigens. Antigen A, antigen B, which are responsible for your blood groups, are self antigens, and HLA, human leukocyte antigen. And now I'll discuss about the human leukocyte antigens later on in the other videos, but human leukocyte antigen is also an antigen which is a self antigen. Your immune system will not produce antibodies against these self antigens, so that's why we are saying that your acquired immunity can recognize which antigen is self and which antigen is foreign or non-self antigen, and your body will never produce antibodies against those self antigens. As this picture is illustrating this, that the self antigen, the immune cells are living happily with the self antigens, but these immune cells are really getting angry over any foreign antigen that comes in your body. Now you must have heard a word called as autoimmune disease. What is an autoimmune disease? If your body starts producing antibodies against your own antigens, that is the self antigens, then this condition is called as an autoimmune disease, which we would be dealing with in other videos. The next and the very most important property of acquired immunity is memory. That means your immune system retains the memory of the first encounter that has taken place with any pathogen or antigen, and will always remember that antigen or pathogen. And if that same antigen or pathogen enters your body for the next time, your immune system would produce a very heightened response against that antigen. I'll discuss it in detail. Let's say there is a foreign agent which has entered your body for the first time, so your immune system would have a first encounter with it, and if it ever enters your body again, then that encounter is called as second encounter, or the third encounter, or the fourth encounter. Now the first encounter with any antigen or pathogen is called as primary immune response, and that primary immune response would take time, and the response would be very mild. That means a low intensity response would be produced, and that would be slow, would take time. Your body would take time to recognize that antigen, to process that antigen, and to produce antibodies and specific immune components against that pathogen. But once they are formed, they are always present inside your body, and whenever there would be a second or a subsequent encounter, the immune response that is produced is called as a secondary immune response, and that immune response is really heightened and intensified and is very rapid. Such kind of response is called as anamnestic response. Anamnestic means something which is remembered and then is displayed at a very heightened intensity, then that is called as anamnestic. I'll explain this thing with the graph here. On the y-axis I am taking concentration of antibodies, and on the x-axis I am talking about time. Now at time equals to 0, there is an initial exposure with an antigen. Now this much time is being taken by the immune system to recognize and process that antigen, and after this much time your body is producing a response which is called as a primary immune response. So you can see the concentration of antibodies at the peak of this primary immune response. This comes at this level. So this is the concentration of antibody at the peak of the primary immune response, and after a certain time, whenever there is the secondary exposure, then the immune response that is produced is really heightened. Look at the peak, look at the concentration of antibody here. Now in this case, this much amount of antibody is being produced. That means the response is really intensified, and plus the response is taking lesser amount of time. Now in this picture, this much amount of time is being taken for the peak to be produced, this much time, but in the secondary exposure only this much time is being taken. Plus you can see the slope, and in this case slope. That's why we are saying that secondary immune response is fast and is really intensified. And now this memory retention is the basis or the principle behind vaccination. Now what is the basic funda behind vaccination? Listen to me carefully. What I am doing, I'll talk about the vaccine against Mycobacterium tuberculosis, a bacteria that produces TB. Now what I am doing, I am taking a bacteria called Mycobacterium tuberculosis and I am weakening it. I am reducing its pathogenicity. Pathogenicity means the capacity to produce pathology or disease. I am reducing its pathogenicity, and then I am introducing this Mycobacterium tuberculosis in your body. Now this bacteria will not produce any disease because this is inactive or dead or Attenuated means we have reduced its potential. It won't produce any disease, but it is a foreign antigen for your body, and your body would produce antibodies against it. That would be primary immune response. And to any vaccine, your body would produce a primary immune response which would be mild and would take time, but nothing to worry, because this won't produce any disease. But your immune system has memorized the antigen, and a lot of antibodies are produced. Now whenever there is an actual encounter with the Mycobacterium tuberculosis, then your body is already ready with multiple antibodies and would produce a heightened response against that bacteria or virus. So this is the basis or the principle behind vaccination. I'll be taking an entire video over vaccination. You can see that video, we'll be releasing it soon. Now how does it happen? How does your body or the immune system memorize the things? Now I'll discuss it in detail here. See, there is a first encounter with foreign agent, and in this first encounter the immune system would take time to process the new kind of antigen. It would recognize, it would process, and then some specific immune cells and specific antibodies are produced against that antigen. Though it takes time, but your body would produce it. Now the immune response which would be produced is called as a primary immune response, which would be slow and mild, and any subsequent infection with the same foreign agent, now your body is already prepared with all those specific immune cells and specific antibodies, and would produce a really heightened and rapid response against that pathogen, which is called as a secondary response. So that's how your memory of immune system works out. I'll be discussing it in detail when I'll be discussing about cell mediated and humoral immunity in our upcoming videos. So let's now discuss some differences between the primary immune response and secondary immune response. The things we have already discussed is that in primary response is always against the first encounter with an antigen. Secondary response is produced in the subsequent encounters. Primary response has got a slow onset, secondary has got a rapid onset. Primary has having a mild low intensity against antigen, but secondary response is having a strong and a high intensity response. Now the most important thing is the type of antibody that is playing a predominant role in this. In the primary immune response, the IgM is having a dominant role, but in secondary response the main antibody responsible is IgG. I'll explain this thing by a simple graph. In this case, see, this is a graph that is being plotted with the blood antibody level on y-axis and days or time on the x-axis. Now see, this is the first antigen exposure, and the first time when there was an antigen inside the body, then your body is producing some concentration of antibodies. This dark brown graph, the line is of IgM. Now after the first antigen exposure, after a few days you can see IgM in your blood, and this IgM is increasing in your body, and it has attained a peak, and after some time it starts reducing, and when IgM starts reducing, the IgG has already started increase, and it would attain a peak after IgM has started reducing. So that means in the primary response the main antibody is IgM, because it is producing peak earlier than IgG, and it has started developing before IgG. But in secondary response, look at the graph carefully. Even at the secondary response, the concentration of IgM is same at the primary response. That means in primary response and secondary response there is no difference in the peak of IgM. But look at IgG. The concentration of IgG has increased multiple folds in the secondary response. This thing proves that IgM is dominant antibody in primary response and IgG is dominant in secondary response. Now I'll be discussing the cells which play roles in acquired immunity. Basically, two types of cells play very important roles in acquired immunity: lymphocytes and antigen presenting cells, APCs. Now what are antigen presenting cells? These are the cells which recognize the antigen and then present those antigens to the lymphocytes. Now lymphocytes will fight against those antigens. You can learn this thing by a simple example, like see, there is someone who has been arrested by the police. Now the police would arrest the police would arrest that thief, and would present it in front of the coat. In the same way, antigen presenting cell would present the culprit, the antigen or the pathogen, towards the lymphocyte to lymphocytes, and antigen presenting cells would process and present the antigens, and the examples are macrophages. And lymphocytes are of two types, T cells and B cells. T lymphocytes or T cells or thymocytes, and on the second type is the B lymphocytes or B cells. Now if I talk about T cells, see both of them T and B cells are lymphocytes, and lymphocytes are WBCs, and WBCs and all other blood cells are produced in bone marrow. So lymphocytes are also produced in bone marrow. So both T lymphocytes and B lymphocytes are produced in bone marrow, but their site of maturation is different, and hence the name. T lymphocytes mature in thymus and B lymphocytes mature in bone marrow. You can remember this by their initial name, T cell. T means thymus, and B cell, B comes from bone marrow. Now B cells are called as B cells not because of bone marrow, there is some other fact behind this, but T cells are called as T cells because of the thymus only, and that's why we are saying thymocytes, and that means cells of thymus. Now T lymphocytes produced in bone marrow but matures in thymus. B lymphocytes are produced and get matured both in bone marrow. If I talk about the concentration, 75 to 80% of lymphocytes are T lymphocytes, but only 10 to 15% lymphocytes are B lymphocytes. If I talk about the function, the main difference is the type of response they produce, the type of immunity they render to you. T cells always provide you with the cell mediated immunity, an immunity which is occurring with the help of some cells, so it is called as cell mediated immunity. But B lymphocytes performs the role of antibody mediated immunity by producing some antibodies. That means B lymphocytes produce some antibodies, and these antibodies are responsible for the immunity. So this is called as antibody mediated immunity, or they are called as humoral immunity. Why humoral immunity? Because humor, as we discussed in our last lectures, means fluid, and all antibodies are present inside the blood. That's why the immunity which is provided by the antibodies present inside the blood is called as humoral immunity. There are certain types of T cells: cytotoxic cells, helper cells, then suppressor cells, and then memory T cells. And B cells are of two types: plasma B cells and memory B cells. Now what are the structures, the functions, and how they perform the role of the immunity? All these things would be discussed in our subsequent lectures. Right now we are talking about only the types of T and B cells. Now the question is, why T and B cells are called so? We have already discussed about T cells. T cells are called as T cells because of thymus. But if I talk about B cells, as I told you, B cells are not called as B cells because they are produced in bone marrow. First I'll tell you the location of thymus. Thymus is present in between two lungs, in front of the trachea and behind the sternum. B cells, the word B has originated from bursa of Fabricius. First of all, the B cells were discovered in a special organ present only in birds, which is called as bursa of Fabricius, which is associated with the alimentary canal of birds, and this is the source of B cells in birds. That's why they are called as B cells. But just to remember the differences, you can say that B cells are produced in bone marrow and T cells are produced in thymus. Next, I'll talk about types of acquired immunity. This would be a very important discussion, so listen to me carefully. See, acquired immunity is of two types: active immunity and passive immunity. As the word implies, active immunity, an immunity in which your body plays an active role, is active immunity, and a type of immunity in which your body performs no active role is called as passive immunity. It would be just a gift to your body. Your body is not doing nothing for this. It is being gifted. It is being gifted. If immune products are gifted to you, gifted to your body, then that comes under the passive immunity. And if your body itself produces those immune components, then it is called as active immunity. I'll talk about the examples. Active immunity would be again of two types: natural and artificial. In natural, the body itself is producing those the entire effect. Like any any infection against a pathogen would produce immune response, and that immune response is a natural active immunity. But now listen to me carefully. As we discussed earlier, the basis of vaccination, in vaccination what we did, we introduced a dead, attenuated or a weakened pathogen into your body. Now your body would elicit an immune response against it. Now the body is actively participating in vaccination. Your body is actively participating. What I have administered in your body, I have administered dead pathogens, but the immune products are formed by the body. That means vaccination is an example of active immunity. Most of the students get confused whether a vaccination is passive or active, just because they think that in vaccination we are providing something to the patient, but analyze it properly, we are providing antigens or pathogens against which immune response is actively produced by the body. So the vaccination is an example of active immunity and not passive immunity. Now I'll talk about passive immunity. It's again of two types, natural and artificial. Now in natural immunity, in both the cases your body would be gifted with the immune components. In natural immunity you would be gifted natural immune components, and in artificial you would be gifted with artificial products. The best example of natural immunity is colostrum. What is colostrum? Colostrum is the initial milk that is produced in the initial days of lactation after delivery, and that is very much rich in IgA. Now the infant that is getting the colostrum from the mother, the body of the infant is doing nothing. The body is not actively participating. It is the IgA is gifted with colostrum by the mother. That's why it is a passive immunity. And the other example is the IgG, which is received by the fetus from the mother. In that case also IgG, immunoglobulin G, a type of antibody, is being gifted by the mother to the fetus. That's why it would be again an example of passive immunity. Now if I talk about artificial immunity, I'll take a case. Let's say there is a patient of tetanus that has been admitted to hospital. Now to treat that patient, what we do in medical field, we administer the patient with preformed antibodies against that antigen of tetanus. So what are we doing? We are administering already formed antibodies. That's why it would be a passive immunity. And anti-tetanus serum, that particular thing is called as anti-tetanus serum, which has preformed bodies against tetanus antigens. And the other example is anti-diphtheric serum, which is called as ADA, which has got preformed antibodies against the toxins of diphtheria. As you can see in the picture, this is an anti-tetanus toxin. So this is an artificial type of passive immunity. So the point to ponder and the point to remember is that the vaccination is an example of active immunity and not passive immunity. Now let's take differences between active and passive immunity. You can see the first figure, a baby is being administered with the vaccine, an example of active immunity, and in the second figure I am administering already formed antibodies into a patient suffering from a disease, and this antibody is then used by the patient to fight against the antigen that is present inside his body, and that is a passive immunity. Active immunity, because it is produced by the body, so it would take time, it's a slow process. But in passive immunity I am directly administering antibodies. In active immunity your body takes time to produce antibodies, so it is slow and takes time. But in passive immunity you are directly administering antibody, so it is a very fast and rapidly produced response. But active immunity would last for a long time because it would produce memory cells, and in passive immunity it would last for a short time. There won't be any immunological memory in passive immunity, but an immunological memory is produced in active immunity. And active immunity will not work in immunodeficient people. What are immunodeficient people? Or the people in which immune system is not working properly. And the best example is the AIDS patient or the HIV patients. As we know, HIV virus acts against directly the immune system. So in such cases immune system is not working properly, so active immunity won't work properly. But in such cases passive immunity has got a very helping role. So thanks a lot for watching us. Keep connected with us. I'll be dealing with our next topics, that is cell mediated immunity and humoral immunity, in the next lectures. Thank you very much for watching us. You can ask any doubts, or if you are having any suggestions for us, you can come and comment in the comment section. Thank you very much for watching us.